Welcome to Scigest, your fortnightly science from the Bioeconomy Science Institute.
Tēnā koutou listeners, no mai haere mai. Welcome back to Scigest. I'm your host Jay Jayaraman, a scientist in the newly formed Bioeconomy Science Institute. You know, we spend a lot of time looking at what plants do on the surface, how they grow, how they taste, or how they look in a field. But today, we're going deep inside the plants themselves. Our guest today has spent her career uncovering the microscopic powerhouses that live within plant tissues. Dr. Linda Johnson is a principal scientist and science group manager in the Bioeconomy Science Institute here in Aotearoa. Some of you may know her already from her and her team's extensive high-impact work over at Ag Research that won them the Prime Minister's Science Prize for 2024. Linda and her team are the matchmakers of the microbial world. They work with endophytes, microbes that live inside plants in a symbiotic relationship that can literally change the biology of the plants, making up our pastures and crops. From pest resistance to climate resilience, it's all happening on a cellular level. Linda, welcome. It's great to have you here.
Hi, thanks for having me. It's a pleasure.
To start us off, for those who haven't spent their lives looking through a microscope, how do you explain the secret life of an endophyte?
The secret life of an endophyte is indeed quite secretive because, first of all, you need to use a microscope to even see that it's there. So the system that I've spent my career working on is very valuable perennial ryegrass plant. It's the most valuable plant species in New Zealand. And inside it is an endophyte. You can't see it. You need to use a microscope. And it basically grows in the intercellular spaces. So these are the spaces between cells. And the only way to look at it would be to peel off a bit of the leaf and put it under a microscope and stain it with something that will show those fungal hyphae. So we have spent a long time trying to understand how these endophytes live with their plants hosts and what do they do.
Okay, so deep inside the plant. So why are these important? Why do you study them?
In the 1980s, we discovered that there was an endophyte called epicloe that is inside the perennial ryegrass, which is the plant species that's used in our pastoral system. And it makes a secondary metabolite, which is a compound, and it can cause these animal disorders. And in fact, the most famous is ryegrass staggers. So sheep will be staggering around. It looks like they're drunk, but because this compound called lolitrin B causes the tremogen, the animal responds by having this disorder. So farmers wanted a solution to that because, you know, in the hill country, sheep can break their leg and it's a really massive animal welfare issue. Plus, there are other impacts on animal health and welfare, including things like dagging and just loss of productivity from that animal. So the first solution or obvious solution would be to remove the endophyte. Once we've removed the endophyte, you can cure the endophyte from the plant and have endophyte-free perennial ryegrass. We found that the grass just didn't persist.
So this is like treating it with a fungicide or something like that?
Yeah,
okay.
Absolutely. Okay. So having these endophyte-free pastures, they don't persist, and we still use that material as a control these days so that we can see the difference. But what endophytes we discovered do, yes, some of them make these mammalian toxic compounds, so they cause livestock issues like the ryegrass staggers, or even heat stress as well is another thing which occurs more often in cattle. The endophyte is there, and it's really important for providing protection against insect pests. So in New Zealand, we have a unique circumstance. We've got our own endemic pasture pests or insect pests. Plus, there's introduced pasture pests such as Argentine stem weevil from Argentina, black beetle from Africa. So we just have this major issue with pasture pests. So there are 6 really important ones, and without the endophyte, they just get eaten up, basically. It's food for insects. So they have a party.
Yeah, so it sounds like the farmers are sort of caught in a catch-22. You want to get rid of it to prevent damage to your animals, but if you get rid of it, it gets damage from all animals, and so you don't have any grass left. How do you navigate that then?
So basically, this started the Endophyte Discovery Program, which we won the Prime Minister's Science Prize for. So what we tried to achieve was to find endophyte strains that still provide the insect protection, which is desirable, but don't produce the animal toxins. So how did we go about that? Well, we needed to find new endophyte strains. So diversity is important, and people think about diversity in plant species, but diversity with microorganisms is actually just as important. And this is an example where we were able to tap into the untapped diversity of these endophytes from finding ryegrass germplasm or seed containing these endophytes from all around the world. So we actually have done bespoke collection trips where we target locations in the world where we might not have been before, or perhaps those plants are trying to survive in a tough environment. We also work with other major germplasm centres and go on collection trips with them, and we share the germplasm. Some comes back to New Zealand. Ag Research hosts the Margot Ford Gene Bank, and we have New Zealand's largest collection of forage seed. So this is a great opportunity for us to look at the endophytes in this seed, which is also very diverse. So what we've been able to do is characterise the endophytes by looking at, really a number of ways. So we started by using something called simple sequence repeats, which enables us to look genetically at the diversity. Nowadays, we can also do genome sequencing. So we look at the diversity. Have we seen that endophyte before on kind of a broad, large scale, if you like? We also look at the chemistry, particularly the compounds that cause these animal toxicosis. So two examples are lolitrin B and ergovaline. So we screen for those, and we also look at the earlier compounds in that pathway, because these are really complex biological pathways. And we make a decision, do we want to progress with that endophyte strain or not? So have we seen this endophyte before based on a genetic diversity screen? We understand its chemistry, and we then will put it into a common background plant line, and we do some entomology or insect screening studies. And we need to ensure that these endophytes provide protection against the major pasture pests that are present in New Zealand, because its job is to protect the plant. So we need to make sure that we're finding endophytes that do that. So going back in time, back into the 1980s when this problem was first discovered, and we found actually it was an endophyte that was making lolitrin B, which caused ryegrass staggers. And then we tried to remove the endophyte and see what happened, but we didn't have persistence of the grass. So the grass just dies because the insects are eating it, having a great time. So then we just started the discovery program, and the first cab off the rank was AR1 in 2001. So AR1 was important because it didn't contain any of these known compounds that cause animal toxicosis. So that was great. And it provided...
So AR1 is a strain of Epicloe?
It's a strain of Epicloe,
okay.
Yes. Yeah,
right.
Commercial strain. And it was sort of the first one that came out of this discovery pipeline. And AR1 was important because it was the first one that we could say didn't cause any livestock disorders, was safe for farmers to use. And it also provided protection against the first major pasture pest that we were aware of back then, which was Argentine stem weevil. So it had the compound pyramine, so it was really important, and it was adopted so rapidly by farmers because they could see this provided a solution. No more ryegrass staggers. We've still got our pastures can survive because they're not getting eaten up by Argentine stem weevil. However, once you get rid of one insect pest, you discover, ecologically, there are other pasture pests as well. And so that then continued the journey of us looking for improved endophytes that could provide protection against many insect pests. So AR1 does provide protection against 3 of those 6 major pasture pests. And then in 2007 was really the breakthrough with AR37. And AR37 is such a critically important endophyte We know that it improves the dry matter production at a minimum of 12%, so it's really great for that plant productivity. We know that it controls really significantly well 5 of the 6 major pasture pests. It doesn't control grass scrub, which would be amazing because that is a real problem in New Zealand pastures, and the chemical that's being used to control it won't be available in the future. So that is going to be a real problem for farmers. AR37 has been inoculated into many different cultivars, and it's the major endophyte that's sold in grass seed, in proprietary ryegrass seed. So it's fantastically important even now in 2026, although it was commercialised in 2007. It's still the best endophyte out there. Recently, it's been replaced, or it's starting to be replaced with AR128, that strain of endophyte. It's very similar to AR37, but it survives, it's more compatible. So this is another whole story really around compatibility of endophytes. So how do they live in their host? And so this other endophyte actually is happier in the grass. It can get into more seed, and it actually stays viable in the seed for longer.
So how do you get an endophyte into a grass? So we talked about the fact that the endophyte is growing within the tissue itself. Do you just spray the seed and it just finds its way in?
I wish. So these are obligate endophytes, which means that they aren't free living in nature, so they only live in their plant host. But luckily, we can get them to grow in a Petri plate on some media. And so what we do is, to get them out first, to know that they're there, you can sterilize a piece of plant tissue. They're mostly found in the very base, and you can put that on a Petri plate and the fungus will grow out. And that enables us to do a lot of work, to be able to work with it in a lab. So that's how you can get the endophyte out. But how do you get the endophyte in? That's actually quite hard, to be honest. And so what we do is we've done a lot of science to actually understand how the endophyte grows, and that's helped as well to understand how to manipulate these fungi and how to actually get them in. So what we do is we germinate a ryegrass seed, and we actually elongate it so the meristem, which is the growing part of the plant, ends up a bit further from the base. And what we do is we take a cut, we cut the meristem just a little bit and take a bit of that endophyte that's growing on a culture plate, and we just literally stuff it in that meristem. It's very crude, and we actually have one of our scientists in the team at the moment who's looking to see if we can develop methodology using a tattooist pen to see if there's going to be a way that we could roboticize this process, because it is a manual process and you need to do it under a binocular microscope. So one of our technicians who's been doing this for years, that takes new endophytes and puts them into cultivars, she's an expert, and like in an hour she could inoculate 100 plants, but it's still a very tedious process. And to be honest, I was really bad at it. You need a lot of experience and very steady hands. And so then what happens is that endophyte will then grow into each new tiller. So in a grass plant, each of its units that you see is a tiller, and it's got this stem. It's really a pseudostem, and the endophyte will then colonize those tissues, but it does so in a really interesting way that actually breaks the dogma for how fungi grow. So fungi, typically, they grow from their tips, but this endophyte, what happens is it gets in between the plant cells and it actually attaches to a plant cell. And as that plant cell grows, it expands. And as it expands, the endophyte, which has these long hyphal tubes or filaments, they expand with it and then put down these new septa, which is like these compartments within hyphae. And then when the plant stops growing, the fungus stops growing as well. And that's how it's a good symbiont or good partner, because it doesn't overuse the resources, because the food it gets from the host plant. So it, like fungal pathogens, for example, they grow in places the plant doesn't want them. They continue to grow. They consume what the plant has. But a symbiont has to very carefully manage its growth inside the host plant.
It doesn't overstay its welcome, basically.
No, and moreover, its growth is synchronized with that of its host plant, so it's really unique. And that's what makes it a good symbiont. But it also means that sometimes you can't take one endophyte and put it into a different cultivar because there might be some issues with how it is growing, because it is a really complex biological process.
So it sounds like it's really host plant species specific, is what you're saying. Is that right?
So these epichloroid endophytes, they have a very narrow host range. So we've sometimes found if you take an endophyte out of, say, a Moroccan grass and we try and put it into a perennial ryegrass, or you take an endophyte from tall fescue and put it into a perennial ryegrass, we might be able to inoculate that plant, but then we find that maybe the endophyte isn't so happy, and so eventually the host plant might kick it out and it becomes endophyte free, or we only get that endophyte colonizing into some of the seeds. And in some cases, back in about 20 odd years ago, we wondered, could we put endophytes into cereals? So, wheat is a really high value crop. Could we protect wheat with these endophytes? And if we took the perennial ryegrass endophytes and we put them into wheat, we couldn't even infect, zero. And so then we thought, well, maybe we could try looking at other endophytes. And in fact, we had this idea that because wheat belongs to this Hordeae tribe, or the Triticeae tribe as it used to be known, and ryegrass belongs to a different tribe, if we could find epichloroid endophytes that fall within that same tribe, maybe there won't be such a division between that compatibility. They'll be closer.
Yeah, they've co-evolved most likely, and therefore
Indeed.
They're better friends and can grow more closely together. Right.
You're dead right. So we've actually had success with putting endophytes into cereals, but we've managed to obtain these from a number of different sources. Alamus grass, which is a very common grassland species in China, for example. We've done a big collection and we've had success with taking those endophytes and putting them into wheat. But in some of the early days and putting them into some modern day cultivars, we found that these endophytes sort of acted like pathogens and stunted the plants, which was really fascinating biological problem. And then we, in a collaboration with a lab in Japan, we were able to take their different lab varieties of wheat that had different substitutions and additions of chromosomes, some of those that came from more wild relatives of wheat. And we found that we could form like what we'd call a compatible interaction where the endophyte wasn't negatively impacting the host plant too much. And so over time, we've been able to actually breed for kind of like trying to do coevolution over a short period of time for a more compatible interaction. And we are now working with the University of Adelaide in Australia, and they've been back crossing our material back into modern day wheats. And recently we've had a wheat breeding company who've said, yeah, we're really interested. We're going to do this work as well. So the reason that they're all interested is because we've been able to show that these endophytes in wheat can provide protection against some of the major fungal diseases of wheat. And it's fantastic control. So it's very, very exciting.
Yeah, this is just staggering. So you're talking about breeding fungi like we do plants,
Yes.
crossing them, selecting individuals that are beneficial.
Yeah.
And we talked about, these endophytes protecting against insects and other pests. But you're saying that it also protects against other fungi, like pathogenic fungi.
Yes.
That's absolutely wonderful. I mean, what a multi-tool for the industry.
It is. And the nice thing about it is because these endophytes get into the seed, they actually get into the embryo of the seed. They're transmitted really well, and you don't have to do seed coating to get the endophyte on. I mean, you do need to have that compatibility sorted so that they can get into each individual seed on a floret. And then what we find is that, well, I guess the holy grail is what we do with our perennial ryegrass is you just sell the seed to farmers and they sow it, and that's all they have to do. So it would be really fantastic if the grain from wheat would have this endophyte in it, and when those plants grow, it grows with the plant. It produces its secondary metabolites or other mechanisms because they can also modulate the host in different ways, plant defenses, all sorts of different things, and protect the plant naturally.
So it looks like you're chasing a bunch of traits. So the chemical trait of protection and then the biological trait of compatibility with the host. Is that something that you're trying to breed? You take a strain of endophyte that is able to produce all the chemicals that you want, but is really fussy about the host, and you cross it with one, let's say, that's really friendly with the host, but is completely useless, doesn't produce any useful chemicals.
Well, yeah, that's sort of the future, I suppose. We've also done a lot of work trying to understand what are the drivers of compatibility, and there's these small proteins known as effectors, and they're important for plant microbe interactions, plant fungal interactions, and what they do is they can allow growth of that fungus inside a plant, whether it's a good thing like a fungal symbiont or an endophyte, or a bad thing like a pathogen. And we've done some work and found that there are these effectors that help the fungus grow in plants. And I guess if we could understand more about the genetics on the host side and on the microorganism side, there are tools that you can use to genetically help with your breeding rather than just looking for a phenotype. So you could go, okay, let's do some crossing. What's happened to the endophyte? Is the endophyte there? Yes, yay, nay. But if you could do it at the genetic level, it would just be so much more rapid. So there's still so much more to understand about endophyte host compatibility, and we have made some interesting discoveries around some of these important effector genes, which we know perhaps stop some of the plant defense responses, allowing that endophyte to live there.
Yeah, I mean, you've touched on something that's very close and dear to my heart, which is effectors. And I work on bacterial pathogens and their effectors, and also on beneficial bacteria that also have effectors.
Yes.
And so what you're trying to do effectively, pardon the pun, is to thread the needle then, right? Is to have enough effectors to not have the plant reject your endophyte, but not so many that work so well that it turns the endophyte into a pathogen.
Absolutely.
Because a really successful endophyte is a pathogen.
Yes. Yeah, that's right. And actually in nature, so the endophytes that we're commercializing, they're asexual endophytes. This is difficult to explain, but it basically means that they're trapped in their host plant and they can't get out. They don't produce any of the sexual spores to enable them to spread from plant to plant. But in Europe, for example, there's epichloroid endophytes, and they can actually choke the developing flowers or the inflorescences. So they form this stroma on the outside, and it's a whole pile of fungal mycelium and hyphae and
yeah, well, that's not so much of a problem for ryegrass unless you're trying to produce seed. But if you're trying to get a wheat endophyte, oh my God, that would be bad.
Yes, that's right. So there is some research that's been currently done trying to understand what is it that enables an endophyte in a host to actually cause that development of a disease symptoms, really. And I think in the asexual endophytes that we've been working with, those genetic elements aren't present. So, that's nice.
Yeah, I mean, that opens up a whole new door, doesn't it? Because, you can work without knowing the genes in the breeding process, like we just discussed.
Yes.
But once you know the genes, that opens the door to gene editing. What are your plans for that?
We are in the last 6 months of a 7-year MB partnership program, which was designed to develop a pipeline for gene editing of these epichloroid endophyte grasses. So as soon as CRISPR technology sort of became available, we really wanted to see if we could use it. So we have been doing genetic modification for a long time to do functional biology to understand the function of genes, because we're trying to explore those compatibility issues and we're trying to find new compounds as well. So often it's easier to start with looking at the genes and the genome. But when CRISPR came along, one thing that had been really frustrating us is that these commercial endophytes like AR37, we couldn't do traditional genetic modification to understand about gene function. And so we knew that there were these interesting clusters of genes that likely encoded these bioactive compounds. But we couldn't do any work to actually test the theory. And so what you do in molecular biology is if you discover this gene cluster and you know that some of these genes, by using bioinformatics, encode these compounds, you would knock that cluster out, and then maybe we do some insect tests and go, okay, that gene cluster is responsible for providing that compound that causes whatever it is. And so with these commercial endophytes, particularly Aotearoa that provides resistance to 5 of our major pasture pests, we don't really know a lot about what those compounds are. And when CRISPR came along, we were really keen to see if we could actually start to knock out genes so we could start to investigate that. The first thing that we did, though, the program was really focused around this pathway which ends up producing a tremogenic compound. These are known as these epoxyxanthotrim compounds. They do cause tremors, but they're very short duration and they're not very high intensity. And you don't actually see the production of these tremors very often on farm. And farmers can manage it by putting their animals on a different paddock. But it's still not ideal, right? Because it still causes an animal health issue. And we thought, well, could we remove those compounds, but what would happen from an insect pest protection? So we were able to get CRISPR-Cas9 to work in these commercial endophytes, which first was like for us a big breakthrough, because then suddenly we had this big tool set that we could use to start to understand how does AR37 do its amazing things, and then could we use that knowledge to find other endophytes, because over the last 20 years, we've come up with one new endophyte to replace AR37, and it's been a real challenge. So knowledge underpins a lot of your discovery. So that's why we were so excited. Okay, we could knock genes out and find out their function. But going back to this complex pathway that ends up making these epoxyxanthotrim compounds, what we've been able to do is actually not only understand this pathway and all the steps along the way, but also been able to investigate what happens when you get rid of those toxic compounds. Now, we do know now that those toxic compounds that cause ryegrass staggers, they also contribute towards some insect pest protection, but only for one of them, and that is Purina. So Purina is an endemic pasture pest. So that is a bit of a shame, and that's one of the amazing traits of AR37. It is one of few endophytes that do provide Purina resistance. However, there are 5 different related species of these epoxyxanthotrim compounds that just change in their decoration of the major compound, and one of them is less tremogenic but still quite bioactive. So we've been able to create this one that will still provide some Purina protection, and probably in the field, maybe no toxicosis. So that work is to be decided. But we've also been able to build up some of these intermediate compounds, and they aren't causing any staggers at all. So we know that that's quite safe. And moreover, we can remove the entire pathway, and that's really helpful for us to interrogate what else that endophyte is able to do. So we can look at removing other secondary metabolite pathways and understand their function. So that work is ongoing, which is very exciting, and I think we're on the way of discovering, well, we have discovered some new compounds, which is great to add to the literature. And we are also learning how to insert genes. And so
this is using gene editing
Using gene editing as well, yes. And so that holds a lot more interesting promise in a way, because yes, it would be a genetically modified organism, but you can add new functionality to the endophyte. So if you think of the endophyte as a surrogate delivery system for delivering traits that you want, and because it's a microorganism, it's a lot easier to do genetic manipulation with than, say, a plant. So what we've been able to do is we have developed CRISPR technology to insert small genes, and the next step is to insert larger genes. Now, these genes we're talking about encode for compounds that are bioactive. So these are secondary metabolite compounds.
And not native to epichloi or other endophytes?
No, not necessarily, but we're also interested in that as well. And so I'll talk about that in a moment. But in this new program that is going to be funded by, or that is funded by the Ag Emission Centre, it's only just starting, and it's in collaboration with the University of Victoria from the Ferrier Institute and the University of Canterbury. So it's a three-way collaboration, and it involves grasslands technology as well. We are working collectively, so we're going to see if we can find some new methane inhibitor compounds and insert those into commercial endophytes. And so as a surrogate delivery system, that endophyte can make things, right? And in this case, methane inhibitors, they will be expressed to a reasonable level in ryegrass. The animal will eat ryegrass, and it will have a methane inhibitor delivered in every mouthful.
So the cows and sheep will burp less methane.
Yeah.
And effectively, you're working towards a climate change solution then.
It is. So that is, a very big promise with a lot of hoops and loops to go through. But basically, CRISPR technology is one of the big enablers for us to do this. And it has to be in commercial endophytes that already provide that pest protection that's required. The other thing of interest to us would be to put in, for example, the lowling pathway. We've not been successful taking these endophytes from torfescue and putting them into perennial ryegrass. We have been able to get them in. We have been able to get them to make lowling compounds. Lowling compounds are really interesting because they provide protection against grass grub, which is the only insect pest that we've not been able to target. And they also provide protection against many pasture pests. They're a really interesting compound that holds a lot of promise. And in New Zealand, we mainly grow in our pastures perennial ryegrass.
Right.
So because of the compatibility issue that we couldn't get enough of those compounds, and then moreover, we couldn't get the endophyte into 100% of all seeds. And when you start to do seed production, if you don't start with every single seed infected by the endophyte after you've done a few rounds, you'll be below 70%. And to sell a commercial product, you need to have 70% infection. So eventually that process, we had to give up on that as an option. But now with genetic technologies and CRISPR-Cas9, we can insert genes. We would like to insert this lowline pathway into our compatible perennial ryegrass endophytes. But there are still technical difficulties with inserting large genes using CRISPR, and there are other potential ways, including CRISPR, to insert genes. And so we need to develop that methodology, and that's part of what we're doing right now.
Yes, and of course you'll need a legislative change to use an organism
Yes.
that has been modified this way.
Absolutely. Yes, we do need legislative change, and my understanding is that the Gene Tech Bill is in the House, but it's stopped at the second reading. So we don't know what the future is for that, but as you know, it takes quite a long time to develop science ideas into products, and so potentially there's still opportunity for us and hope that in the future New Zealand will change its stance on genetic technologies.
Okay, going back really quickly to the endophytes themselves, we've talked about pest resistance, we've talked about methane production resistance. Are there any other traits that you can think of that endophytes can provide to their host plants?
Well, so today I really just talked about epichloi endophytes of grasses, and we've predominantly deployed them for their protection against insect pests. But that doesn't mean that's the only thing that they do. We're really interested in climate resilience, and so with climate change, there are going to be a number of other traits that will be important. So drought tolerance is one that comes to mind, heat tolerance, maybe improving nutrient use efficiency. There's a whole pile of traits that endophytes could provide help for. In our work that we've done with cereal endophytes, we've been able to show that they're really effective as controls against pathogens, so fungal diseases. And we also know that they also stimulate plant growth, so there's that biostimulation aspect. AR37 endophyte is interesting because it actually makes the plant roots grow longer.
Interesting.
So we're actually doing a little bit of work trying to understand how does that impact its ability to withstand drought, because you've got longer roots. And there are, every single plant species has endophytes. And I'm just talking about these really special endophytes that are quite unique, that have a synchronized growth habit with their plants. But there are many other endophytes, and some of them...
So beyond grasses as well?
Yes. And every single... we have a microbiome in our gut. And I think a lot of people know about that and how important it is to have the right microbiome, to have a healthy gut. And it's the same for plant species. They have a microbiome, so a whole pile of different microorganisms. And the ones that are living inside the plants are endophytes. The ones that live on the leaves, they're epiphytes and so forth. So we like to characterize everything as humans so that we can understand what we see. But, the microbiome is really important. And I think in the future we're going to start to see not just these single endophytes or single microbes applied for plant protection, but, a consortia of microorganisms. And that means that these benefits that we want these endophytes or microbes to give to their host plants are wider. So another example is the rhizobia symbiosis, and our pastures are not just comprised of perennial ryegrass, they also have a small percentage of clover, and white clover is a really important species, and we could reduce the amount of nitrogen we use through the rhizobia symbiont, and that really helps with reducing the amount of nitrogen. So that's another example of, it's not an endophyte per se, but it's still a beneficial microorganism.
Very exciting. The future does indeed look very green. And that brings us to the end of today's episode. A massive thank you to Dr. Linda Johnson for joining us and pulling back the curtain on the invisible world of endophytes.
It's been a pleasure. I've so enjoyed talking about our research. Always been very passionate about endophytes, and really they can do so much.
Thank you so much. It's pretty incredible to think that the future of New Zealand's multi-billion-dollar agricultural industry, not to mention our climate resilience, might just depend on these tiny fungal partners tucked away inside a blade of grass. If you enjoyed this episode, we'd love for you to help us grow. Please take a moment to rate and review Scigest on your favorite podcast platform, wherever you're listening right now. Your reviews are the best way to help other curious minds find the show. For more on the work being done at the Bioeconomy Science Institute, head over to our website or follow us on social media for the latest breakthroughs. Thanks for tuning in, and catch you on the next episode of Scigest.